Good afternoon. I'm Lynn Weil, Director of External Affairs with CSET, the Center for Security and Emerging Technology at Georgetown University. Today, we'll hold a conversation on assessing pandemic preparedness and ways to ensure effective response by governments in the future. But before we start, I'd like to do a brief bit of housekeeping. All attendees' microphones are muted. If you're on a computer and you experience any technical issues, use the chat function at the bottom of your screen to alert us, and a CSET team member will try to help you out. Don't use the chat for anything else just yet, and we'll come back to it in a bit after the presentations. And now it's my pleasure to hand the reins over to our moderator. Anna Puglisi is the Director of Biotechnology Programs and a Senior Fellow with CSET. She previously served as the National Counterintelligence Officer for East Asia, advising US and foreign government officials at the highest levels, academia, and the private sector on counterintelligence issues. As a member of the Senior Analytics Service, she developed multidisciplinary efforts to understand global technology developments and their impact on US competitiveness and national security, as well as efforts to target US technology. Anna also started a government-wide working group looking at developments in biological sciences and has worked on several biosecurity issues. She has received numerous honors and awards, including the FBI Director's Award for Excellence. Anna, over to you.
Thank you so much, Lynn. What an exciting couple weeks for bio. We could not have planned this better. Both the Executive Order and CHIPS and Science Act highlight how biotechnology and the bioeconomy are critical parts of national security and key to future US competitiveness. Given the importance of the field, it will be essential for decision makers to understand the current landscape of this research as a way to put developments into perspective when they consider what might be considered risky and also how to be better prepared for future pandemics. New developments in biotechnology create many opportunities for human health, manufacturing, and discovery. But there is the possibility that this research may push societal norms regarding the use of research that enhances virulence or transmissibility, as well as introducing risks and the potential for misuse. As we move forward, differences in approach and regulations could potentially lead to ethical asymmetries where prescribed research in one country or region could be explored in another in ways that could impact the speed and adaptation of technology, as well as US competitiveness. CSET currently has a nascent effort focused on characterizing and mapping worldwide biotech capabilities to create a multi-layered set of signals to detect and understand this research. We believe that understanding which categories of research are being explored by whom and where, as well how is it changing over time, is creating context for decision makers as they grapple with how to promote the development of this exciting field, be ready for future pandemics, and protect against misuse. Today, we are going to hear about viral families and why this is a good approach to pandemic preparedness. We will also highlight a part of CSET's efforts focused on pathogens of pandemic potential and where we are characterizing the current state of respiratory viral family research worldwide. It's my pleasure at this point to introduce Dr. Amesh Adaja. Dr. Adaja is a senior scholar at Johns Hopkins Center for Health Security, an adjunct assistant professor at the Johns Hopkins Bloomberg School of Public Health, and an affiliate of the Johns Hopkins Center for Global Health. His work is focused on emerging infectious disease, pandemic preparedness, and biosecurity. Dr. Adaja is served on US government panels tasked with developing guidelines for the treatment of plague, botulism, and anthrax in mass casualty settings, the System of Care for Infectious Disease Emergencies, and the National Academy Steering Committee for Diagnostic Excellence. He also served as an external advisor to the New York City Health and Hospitals Emergency Management Highly Infectious Disease Training Program, and on a US Federal Emergency Management Agency working group on nuclear disaster recovery. Dr. Adaja's expertise is frequently sought by international and national media, and he is also an editor at the Journal of Health Security and a member of various medical societies. Dr. Adaja, over to you.
Thank you for that introduction. Thank you for inviting me to participate in this webinar. What I was going to do for about the next 8 minutes or so is sort of talk off the cuff about the whole idea of thinking about pandemic preparedness as one of looking at viral families most likely to cause a pandemic and kind of focusing the efforts that way. And in order to do that, I kind of have to give you the origin story of how I started thinking about this and what it was like when I started in the field, which was around 2008 when I was still in training as an infectious disease fellow. At that time, the way that people thought about pandemic preparedness was largely based on the category A agents that were weaponized by the former Soviet Union. Botulism, anthrax, plague, tularemia, smallpox, viral hemorrhagic fevers. That's what people focus on. And then basically they added influenza to that list. And that was considered to be pandemic preparedness. And while there definitely are synergies in terms of public health preparedness, medical countermeasure development, strategic stockpiling, surveillance, public health infrastructure, hospital capacity, it's not always going to be the same. Because if you think about what it is, if you're designing, if you get in the mind of a biological weapons engineer, they don't necessarily care so much about human to human transmission. They care about shock and awe. They care about incapacitating troops. They care about area denial. If you put anthrax spores, for example, in a certain part of a country that might not be habitable. Those are not the same considerations when you think about a pandemic. And that's why, for example, diseases like tularemia and botulism, anthrax were weaponized in some of the more attractive weapons, so to speak, versus what causes a pandemic. And I really wanted to think, is that the best way to think about pandemic preparedness, is to go down that list and check all the boxes on and make sure you have countermeasures for all of those? Would that make us more resilient? And then you tack influenza on only because it caused pandemics in the past. So back around 2015, 2016, I really started thinking about this in great detail. And I wanted to kind of be philosophical about it and totally agnostic and understand, what is it that causes a pathogen to have the characteristics that allow it to cause a pandemic? What biological characteristics have to be present? And I wanted to be completely and totally agnostic. I wanted to go into this, and this is a project I wrote a report on back before the pandemic, around 2016 or so, called The Characteristics of Pandemic Pathogens. And as part of that, I started out completely with almost a blank slate, saying, let's just not assume it's going to be a virus. Let's say, could it be a bacteria? Could it be a fungi? Could it be a parasite? Could it be a protozoa? Could it be a prion? Could it be a helminth? And could it be a virus? And I just wanted to drill into that and say, what is it that you need to have? What's the alchemy of a pandemic pathogen? And you come on to a few different things. Obviously, to cause a pandemic, something has to be able to spread efficiently from person to person. We're not talking about non-contagious infections. And when you think about what's the most efficient way, I think we've all seen this with COVID-19. It's when something is spread through the respiratory route, through coughing, talking, laughing, all breathing. That's very difficult for public health interventions to actually intervene upon. When you think about fecal oral or mosquito-borne or whatever other way, those are going to be very limited. Fecal oral, for example, can be limited with sanitation and hand washing. Something is spread by mosquitoes, it's going to be limited by the mosquito's range. But if something is spread through the respiratory route, it's not going to be able to easily be stopped. So something has to be spread through the respiratory route. Number two, you really have to have the ability of this to be efficient at that respiratory spread. And that means being contagious, for example, during its incubation period, or contagious when people have mild symptoms so that they can socially interact. Number three, it has to be relatively new to the human population, meaning you have to have an immunologically naive population, one in which there's not enough immunity to be able to prevent infection. Four, you have to have no off-the-shelf antiviral, no off-the-shelf vaccine that's going to work. Usually something that has no countermeasures against it, that's going to be likely to be what occurs. And when you kind of map that onto the known group of pathogens, it really comes down to viruses, because bacteria, even the most dangerous and scary multi-drug resistant bacteria, we can usually craft something that will work. And those don't usually affect healthy individuals. They affect people that are in nursing homes, people who are debilitated, immunocompromised individuals, not something that spreads in the general population. Fungus, we know, are limited. Their ability to transmit between human to human is severely limited, and they don't grow well at our human 98.6 degree Fahrenheit filter. And if you go through all of those, it really comes down to, it's going to be viruses. And when you look at viruses, so now we're talking about viruses spread through the respiratory route. Many of them are going to be RNA in origin, because that may help them have a wider host range, infect other mammals outside of humans. And that's not always going to be the case. There are some DNA viruses that that probably have that pandemic capacity. And when you do all of that, you come down to about a half dozen or so viral families. And I think these probably sound kind of second nature now because people are talking about respiratory viruses, but this wasn't the case back when I was looking at this idea. So if you think about influenza viruses, coronaviruses, paramyxoviruses, adenoviruses, those types of pneumoviruses, there's a group of them there that all I think are where the emerging pandemic pathogens are going to emerge from. And that gives you a lot of insight, because that means when you're thinking about all the exotic viruses that are on this planet, it's most likely that none of them are going to be able to cause a pandemic. They may be able to cause an outbreak or an epidemic and some disruption, but they're not necessarily going to be able to cause a pandemic. And I think that what we need to do when you think about pandemic preparedness is use those viral families as a lens to hone the way the government and private industry and the intelligence community, the military think about pandemics, because those viruses are likely, those viral families are likely where the next pandemic will come from. And it might not be the exact thing that you see. So it might not be, for example, let's like coronavirus, for example, it wasn't SARS-CoV-1 that caused the pandemic, but SARS-CoV-2, something adjacent to something that was already kind of out there. So, for example, when you focus on paramyxoviruses, a lot of people think about Nipah virus. The virus that spread from bats has caused outbreaks in places like India, Bangladesh, Malaysia. It's probably not Nipah that's going to cause the pandemic, but maybe something Nipah adjacent, another zoonotic paramyxovirus. And I think that's, at least to me, enlightening, because that tells us that you have to prepare, when you're preparing for a pandemic and you're looking at a viral family, all the work you do in that viral family is going to be useful, because it's going to teach you about correlates of protection. It's going to teach you about common pathways of immune response. It's going to maybe give you leads into medical countermeasures like antivirals or monoclonal antibodies that could be used. And I think this is starting to become more and more accepted, that you hear about the NIH's prototype pathogen approach. CEPI, the Coalition for Epidemic Preparedness Innovation, is also looking at viral families. And I think this is something that will make us much more resilient and better thinking. And it also helps to not confuse things that are viral discovery from pandemic preparedness. Viral discovery is an important task on its own, but it's not synonymous with pandemic preparedness, because if you're looking at viruses that infect salamanders, for example, most of those are not going to be able to cause pandemics or even infect humans. And it's going to be a great viral project to understand what's infecting salamanders, or maybe causing one-off infections in people who handle salamanders. But that's not the same thing as pandemic preparedness. So I think it's important to really hone in on what will actually cause a pandemic. And I think this has lots of implications, because all those respiratory viral families cause infections today. They all, RSV, adenoviruses, paraflu, rhinoviruses, and we don't have a good handle on them. We don't really know how many people get sick with them. How many times have you went to the doctor and you told you've got some virus, but not which virus? I think we really need to take the tools that we have, respiratory viral panels to do diagnostic testing, to get a better handle on those respiratory viral families and what they're doing now. Who are they infecting? Who has risk factors for severe disease? And that will not only give us epidemiology, but it might provide an impetus for companies to think about vaccines for some of these. We know there's an RSV vaccine that's in the offing right now, or antivirals, and all of that's going to help us when maybe one of the cousins of those viral families causes a pandemic. And I think this is one of my biggest take-home points, is that we have to get better at dealing with the ordinary day-to-day respiratory viruses that infect us all the time, because that's going to impact the way we deal with pandemics, and it's going to help us have things on the shelf that could be useful to increase our resiliency. And I think that this should be how we think about this moving forward. And I think SARS-CoV-2 gives us that opportunity to sort of get this right. And hopefully there's enough initiative to do this. But I think that this is kind of where I've come to the conclusion that I think this is how pandemic preparedness would be better facilitated. And I think you'll hear in the rest of the webinar about those viral families and where that research is going on. But I think it is a major priority to understand what's making people sick, what the epidemiology is of these viruses and these viral families, and to start developing countermeasures. And one of the things from COVID-19 is many of these viral families sort of were constrained by social distancing and masking, and we didn't have any of them, but some persisted. We had off-season RSV. We saw rhinoviruses circulating during the pandemic. That itself is interesting because that tells you those are more resilient viral families than others. For example, parts of influenza, some influenza family trees basically went extinct. So all of this, I think, provides a lot of a big basis to really get robust about pandemic preparedness and think about these viral families. And not only, I think, will it help us with pandemic preparedness, but I think it will help us with ordinary care, because I think we do a poor job with respiratory viruses. And those are kind of my opening remarks that set the tone for how to think about viral families and where they all come from. And again, if you are interested, I recommend the report I wrote called The Characteristics of Pandemic Pathogens. If you look that up on the Center for Health Security website, or just Google it, you'll see that and see some of the rationale for how I came to these conclusions. But hopefully that was interesting. Thank you, and I'm looking forward to questions you may have.
Thank you so much, Dr. Adaja. So now I'd like to take the opportunity to introduce one of our fellows here at CSET, Dr. Caroline Scherger. Caroline Scherger is a biotech research fellow at CSET. She received her PhD in molecular medicine from the Cleveland Clinic and was previously an S&T policy fellow at the National Academies of Science. Dr. Scherger, over to you.
Oh, thank you for the introduction, Anna and Dr. Adaja, for your insight into why it is so important to approach pandemics and infectious diseases responses from this perspective. At CSET, we use data to analyze complex policy problems such as where do we need to be prepared for the next pandemic or major viral outbreak? And I want to talk a little bit about that today. To begin to look at pandemic preparedness, our team investigated viral families of pandemic potential that Dr. Adaja was talking about previously. And as shown on this chart, our research highlights that we don't have many medical countermeasures. We found that countermeasures, including both preventative vaccines and FDA-approved drugs, are not equally distributed among the different viruses. And some viral families have no vaccines and others don't have any drugs. And we also have the separate issue of, will these vaccines and drugs that we do have be available when we need them? So we looked at where these countermeasures are manufactured. Altogether, they have limited domestic capabilities, meaning that the US is reliant on foreign sources for half the vaccines and drugs we need. In addition, there's a lack of redundancy in the manufacturing of these countermeasures. So what happens if there's a disruption at the one facility that makes a drug? Would we not have access to them if we need it? So we also looked into how we get to having these countermeasures and what is the landscape of the foundational research and clinical trials that are necessary for developing more vaccines and drugs we use to combat diseases. We found that overall, research publications and clinical trials are reactionary and have a time lag from major viral outbreaks that occurred. For example, both publications and clinical trials on the flu increased in the months following the 2009 H1N1 pandemic. This means that while we know that these viruses can cause serious harm and spread throughout the world, we still have to ask the question, are we currently investing in the foundational research on the front end before these events happen, so we have these medical countermeasures to prevent and combat the diseases? So that's a little taste of our ongoing projects and data on the biotech team at CSET, and I'm really looking forward to our discussion on this. So I'm going to turn it back over.
Great. Well, thank you, Dr. Scherger and Dr. Adaja, for your presentations. They are sure to prompt a lively discussion. Our moderator, Anna Puglisi, will ask the first question to kick off the Q&A. And in the meantime, if anyone would like to ask a question, please type it in the chat. Now's your chance. We may not get to all of them, but we'll do our best to cover as many as we can. Those who are phoning in for this meeting will not be able to ask questions, and we thank you for understanding the technical limitations here.
Great, thank you so much, Lynn. So I do have a question for you, Dr. Adaja. So is the idea of viral family approach, is this well accepted or not accepted in both policy and research communities?
I think over the last year or two, it has become much more well accepted. When I started thinking about this, and I interviewed lots of people, I do a lot of kind of subject matter expert interviews just to get people's take on what these ideas that I had. And a lot of them were saying, well, what viral families? It's just one, it's just influenza. That was directly a quote from an interview. So most people didn't even conceptualize pre-pandemic COVID or coronaviruses as a pandemic threat. Some people did, but others did not. So I think that this wasn't something that everybody thought of it. A lot of people really were wedded to this list-based idea that came from, as I said, the former Soviet bioweapons program, and equating that with pandemic preparedness. Now, as I mentioned at the end of my remarks, the NIH has started this pandemic prototype pathogen approach, which I think has a lot of similarities to the viral family approach. And if you look, for example, at BARDA's funding, they're actually asking for congressional appropriations to fund a viral family approach. So this, I think, has taken off and got a life of its own. And I think it makes sense. And I think it will actually be something that makes us better prepared for the next infectious disease emergency that we face.
Thank you very much. We do have a question on how does this relate to the new efforts on biomanufacturing?
So the most recent executive order, I believe it was about a week ago, was focusing on biomanufacturing and reshoring a lot of these facilities over to the US, which, as our data shows, is extremely needed to be able to have access to drugs and vaccines and not have to rely on importing. A consideration about manufacturing drugs is that they require a lot of specified materials that are hard to track down on the FDA website to determine what is considered an active ingredient or a starting material. And there's also other considerations such as ensuring vaccines are cold or they're able to transport to people who can provide these vaccines. So we're really excited that the White House is taking this as a serious priority moving forward, so we can are able to address some of these gaps that we have found.
Thank you very much. Can you talk a little bit about the research and how that is distributed worldwide?
Sure. So a little bit about how we measured this research. We looked at publications that mention any virus in these specific viral families. And then we queried PubMed to determine the year and where these publications were published from. And we found that overwhelmingly, about half and half publications are from the US and China. So these different viral families have either the US is leading in publications or China. And we also see that reflected in clinical trials. So the US has about 30% of clinical trials registered on clinicaltrials.gov. And using our taxonomy on the viruses that exist in these families, we're able to count these numbers over time. And you see really nice curves go up. It's really cool. After H1N1, it's a peak in publications and a peak in clinical trials after. And we see a huge increase in publications on coronavirus after 2020 that skews the graph a little bit, actually. So it's very interesting to see how the real world affects publication output.
Thank you for that. We do have a question about what kind of therapies are needed to address future pandemics. Is it one kind of therapeutic? Or are they different varieties?
So I'll take this. I think that it's going to be all of the above. I think we're going to need small molecule antivirals that could be used to treat patients and also to even be used as prophylaxis. We're going to need monoclonal antibodies. We're clearly going to need vaccines because prevention is better than having to treat. And then I think lastly, we're going to need immune modulating type of drugs that we saw were so successful in severe cases of COVID-19. So I think it's all of the above is going to be needed. And that also goes for diagnostics as well. So it's really the whole gamut. And as you heard, many of these viral families don't have any countermeasures against them. And when they do, it's maybe you might have a vaccine for some paramyxoviruses, for example, but there's no antivirals for many of these, and really puny amount of vaccines for some of the other family members. So I think that there's a lot of work to be done in those viral families. And this is where I say that if you actually start diagnosing these and know this is the burden of para-influenza virus, for example, in the United States, that may incite a company to say, okay, maybe there is a role here for an antiviral or a vaccine, which would then help us if some para-influenza adjacent issue causes a pandemic, or if a weird para-influenza virus causes a major outbreak that we need to get a handle on.
Thank you. So we have a couple of questions that are asking about, is there a more qualitative way to evaluate what is going on research-wise more than actual papers, given some of the questions about numbers of papers and or the quality of papers?
That's a really great question, as the threshold for publication is different for different scientific journals. And we count papers that are in PubMed, so they're in English and they are publicly available. However, that is one of the only ways that we can look at basic research output in one setting. You can look at grants, you can look at patents, et cetera, as a research output. But to have an unbiased approach where multiple countries, multiple institutions have that kind of information, we looked at pandemics. At publications. But this is also why we're looking at all of these different factors to paint a picture all together of what this data means. So while it's not the perfect indicator, in aggregate, we'll be able to provide a little bit more insight about where the research is going on, but how is that research being translated into clinical trials and then the physical outputs of vaccines and drugs.
Thank you. So we do have a question about, and this kind of gets to how I teed up today's webinar, is how do we sustain the interest of U.S. policymakers in supporting sustained funding for research in this area, especially as interest may drop as we move out of the COVID pandemic?
I think that's going to be the biggest challenge, because obviously we heard about how reactive all the clinical trials are and everything, and that's exactly how the government, I think, responds. And we've seen it with monkeypox itself, very reactive instead of a proactive approach. But I think the viral family approach gives you kind of a roadmap of what they should be working on. And hopefully, and I don't know what, I don't mean to offend any of the Washington, D.C. people, and it's hard to know exactly how they'll approach this, because you would think that after COVID-19 and 1 million deaths, massive economic disruption, all kinds of political turmoil, that they would realize that it's really important to get this right, that it is something that extends beyond election cycles. This is something that needs to be thought of as national defense, and be as nonpartisan as national defense. I don't know if that's asking too much, but I think just showing them how much money it costs to stop something once it's started should be enough. But again, I've given up trying to understand how that all works there. I'm just an infectious disease doctor. I'm much better dealing with bacteria and viruses and sick patients than I am with Washington, D.C. on these things.
And I think it's also important to emphasize that how we get drugs and vaccines is through all the years of labor on research and moving that through clinical trials. I have a little faith in how there's these spotlights on how the COVID vaccine was developed, showing that through years and years of research, we were able to mobilize quickly, because these things don't just pop up overnight. They require a lot of graduate students, PIs, et cetera, to do that kind of work, and they need the funding to do so. So hoping that connecting that to the vaccines and drugs that we so clearly need, and in the stark pie chart, we'll be able to highlight those gaps and think about how can we address them through basic research.
Thank you. We have a question about vaccine development. So given the current state of vaccine technology, is describing the different viral families enough that you can have vaccines that can address the entire family, or is it more complicated than that?
I'll take a stab at this first. I think it's more complicated than that. But the thing is, if you have a viral family approach, that may get people to start working in that viral family and make something for what may be a human pathogen. So, for example, I keep going back to parainfluenza. It's an easy one. So, suppose they make a parainfluenza vaccine. The knowledge that they gain would be useful for any other paramyxovirus that might be a pandemic potential pathogen. And this is actually borne out because the work they did with SARS-CoV-1 and MERS, particularly MERS, was really instrumental in the way the NIH, that the group at the Vaccine Research Center, was able to figure out how to stabilize the spike protein and modify the spike protein of SARS-CoV-2. So that saves you a lot of time up front. Whether or not then you might stumble upon a universal vaccine that takes out the whole family, that would be really, really the kind of the holy grail. But the point is, I think it's even more minor developments in a viral family are going to make people much more comfortable working with that virus family, understanding, as I said, what the correlate of protection is, what they need to make a vaccine against, what the important proteins are to maybe disrupt with antivirals. All of that will happen just by working in the viral family. And it's important also because a lot of the times congressional funding was, you have to work on this virus XYZ, and that's it. And that's what the funding was put on because that was on some list. But now, if you have a viral family approach, you can say, well, I'm not working on that particular virus, but I'm working on viral families that contain pandemic pathogens, including the one that you guys are interested in Congress that you've put a line item for. Can that funding be used that way? And that, I think, is important, especially when you think about BARDA, where they have this emerging infectious disease and influenza division, but they don't really do anything other than influenza, and they're very tied to a specific allocation. This is for Zika, nothing else. If you can say, work on these viral families, and it's all part of pandemic preparedness as well as regular regular research, I think that's a lot more fruitful than having them tied to waiting for a specific appropriation to be able to fund research into what could be an important task for pandemic preparedness.
Okay, so we have several questions focused on kind of the foundational facilities and infrastructure that are needed in order to be prepared, whether that be manufacturing capabilities or even what kind of tools. AI has come up a lot that will help us better design either therapeutics or vaccines in the future.
So one of the problems with working with viruses that are dangerous is that they're dangerous and require a lot of special facilities, such as biosafety level 2, 3, 4, the skills, the workforce, as well as other ways to be able to do that kind of research at the basic level. So you mentioned also, looking at AI and how computational modeling can help. During the initial COVID research phase, people were using protein modeling to determine the spike proteins on the outside and what those conformations are, so we could develop vaccines against those particular proteins. So AI has helped eliminate some legwork. It's not perfect in terms of being able to it's only able to predict certain protein conformations or what there might be able to exist. You still have to do the legwork of proving that that's how the protein works, et cetera. But it does speed things up a lot. And we saw how the world came together to work on that project.
Okay, thank you very much. For these kinds of facilities, what kind of investments are we thinking or we're looking at over time? There's a lot of questions about that. I know that, I guess, the devil's in the details with the new legislation as to how that is actually eventually worked out. So we do have a question on focusing on not only viral families, but the potential for manipulation of viruses, and whether the viral family approach will leave us better prepared for some viruses that might be either enhanced or changed their gain of function.
I think the answer probably be yeah, it will be yes, because if there is manipulation of viruses, it's often going to be those viral families that are most likely to cause a pandemic, whether this is being done as genuine research or whether it's being done for nefarious purposes. They're going to be starting with, some kind of substrate they're going to be already picking things that have characteristics that are useful for whatever purpose that is. And if you look at when there was manipulation in the former Soviet Union, it often were things that were already deadly on their own or had characteristics that were useful from a biological weapon standpoint. And I think the same would be the case for if they were trying to use a respiratory virus to spark a pandemic. So I think it would help us. And I think the more science you do and the more understanding and countermeasures you have for all of these, the better it is. And then I would also say the more of a bioeconomy that we have, because people are interested in making vaccines and understanding this, the more the biotech sector is robust in a country. I think that's almost like all hazards preparedness for any type of biological event, whether natural, whether engineered, or whatever it may be. Because if you look at the story of COVID-19, it's mostly how the bioeconomy was able to really surge to be able to meet this demand, rather than a lot of the traditional public health, which often failed and got mired up in politics.
And I think it's also regarding the gain of function question is the role of the government to provide resources, regulations, like insight, training, et cetera, for these researchers to do these things safely, to find alternatives to answer the same question and not just do research because it's cool. With all that, the goal at the end is to be able to figure out how the flu works or determine the best way to approach designing vaccines, et cetera.
Okay, we have a workforce question. I know a lot of the focus is on PhDs and research, but are there studies or that you're aware of that look at what kind of a technically proficient workforce would be needed for the manufacturing of these potential either therapeutics?
I think that raises the question about the need for people who don't have PhDs but have these technical skills for doing intricate and important work in manufacturing facilities. Is highly trained, but it doesn't necessarily require a PhD. Maybe community college training or these programs to ensure that people are doing it correctly, that we have a pathway for a workforce in the biomanufacturing field.
So we have one more question, another question about the kinds of techniques that are used, and questions about understanding what techniques. The question really relates to synthetic biology, but whether it be genome editing, that kind of categories of research that help us better understand how the viruses work. Can you talk a little bit about that?
I think some of it is just basic virology. It's not necessarily anything fancy. I think as biotechnology has improved, there's going to be newer techniques that can be used, but a lot of it is just simple things like cell culture, animal models, understanding the pathophysiology of what's going on, and then understanding the viral life cycle. And that's been done without gene editing or any of those types of measures in the past. So I don't necessarily think it's anything that's completely 21st century, even though that actually helps them do that faster. But it's just a basic understanding that is lacking. And a lot of these viruses, just a lot less interest, because many of those viral families cause really mild illness and they haven't really been considered a priority. But that was the case with coronaviruses until 2003, that this was a mild viral family that no one really did any basic work on that much. It was because it wasn't that important.
So it sounds like a matter of approach and how we prioritize some of this research. A number of questions related to, in some ways, what would it take to change the culture of what is actually researched and where those priorities are placed?
The biggest incentive, I believe, would be funding and grants, because that is how researchers are able to sustain their labs and their projects, are by showing the granting agency, look, we're doing this research that you asked for. We're tailoring it to what you're requesting so we can receive this money. So I really believe it's at the top where the money comes from to be able to incentivize researchers to follow the basic work in these viral families.
Thank you. We have a question about kind of milestones or indicators. So as research begins to ramp up in some of these areas, how do we know we're on the right path? Like, what would be kind of a roadmap to put some of these strategies in place?
I think at the end, it's going to have to be products, medical countermeasures. And that's what they did actually with Project BioShield and the work on Category A biological weapons, that they had kind of criteria that we bring this many things into full FDA approval in terms of vaccines, 2 antivirals for smallpox, for example. So I think we make milestones like that we have prototype vaccines for pathogens in a viral family. We have antiviral candidates, monoclonal antibody candidates. All of that, I think, would be part of it. And whether they come all the way to FDA approval or they're kind of at phase 2B and then ramped up if there's something that occurs, I think that remains to be seen. But I think it has to be product development.
Okay, thank you. We have a question about within the different viral families, how would you prioritize those of the 7?
I think I think influenza still has to be considered the highest priority threat, no matter how you look at it, because that is the virus that we know can cause pandemics, and we have multiple avian flu species that like H7N9, H5N6, as well as some interesting swine species like the G4 swine species in China that are, I think, of concern. So I think flu has to be probably prioritized the most. And the others, I think you have to kind of parse them. I think that there's probably not one specific way to do it. I think we don't quite know. Coronaviruses are pretty well resourced against right now. And I think there's still work that needs to be done. We need better antivirals and obviously better vaccines. But I think that's probably well taken care of. And I think of the others, I think it all depends upon how you look at it. And I don't know that I've thought about how to prioritize them. I think the RNA viruses that are there are probably more likely. So paramyxoviruses, pneumoviruses, some of the enteroviruses, that those would probably be higher priority to me than adenovirus. And in that list, I initially thought about pox viruses, but I think it's very, very difficult for a pox virus to be able to cause a pandemic in the modern world because we have vaccines against them and antivirals against them and diagnostic tests. So I think I would probably prioritize the RNA viruses if I were to pick, but I think it could be argued either way.
Great, thank you. I have one additional question on kind of the workforce. What is the ideal background for someone wanting to move into this kind of research? I notice you have a very multidisciplinary background.
If that's for me, I'm not a bench researcher. I'm not a person that does that type of work. I think a lot about these things, and I try to be able to understand all that research. And then I mostly see patients and think about things from a clinical perspective, which maybe helps me think about pandemic pathogens a little bit differently than others do. So I'm probably not the best person to tell you what the research, the wet lab research background that you might need to be able to do that. So I'll defer to Caroline to do it.
Yes, I grad school was a lot of wet lab experience, holding a pipette. There's a lot of barriers to graduate school as they become more competitive. Having a background in biology, chemistry, biomedical engineering, and these sciences for undergraduate degrees, and then either pursuing a master's before a PhD. But there's also other people who work in labs that aren't coming from that PhD program. We have MDs. We also have veterinary technicians who work with the animals and animal models, and they are absolutely critical to helping develop more of those translatable experiments. So also the ones that we're interested in, because if something works in a mouse versus a cell, that might have more promise. So there are all these different components that make up a lab. But I also think that my experience in the lab has helped inform how I view policy and how science is funded and how we need to work through basic research to make these medical countermeasures. I'm not sure if the question was more of the wet lab experience versus the policy research experience.
No, I think it was more of the wet lab experience. But I'm going to pull a thread because we do have a question about this as well. Is the role of animal models and the importance of those? Can you speak to that?
Sure. So things can work in a cell in the petri dish. That's a very controlled environment that you don't have blood pumping, you don't have other environmental factors that can interact with a drug or vaccine. Making something work in a human body is extremely difficult. You have, the pH of your blood to worry about. You have to worry about drugs being metabolized and not even making it to where they're trying to get to. And animal models kind of help bridge this a little bit. So mice are a really easy animal model to work with because they, reproduce quickly. They have, after the Human Genome Project, we understand genetics of mice models. But another important model I wanted to talk about real quickly are non-human primates, which are really close to humans and therefore a helpful model when testing the safety and efficacy of drugs and vaccines in a humane way. But non-human primate models aren't being used as frequently. There's no availability in the US as much anymore. So a lot of experiments and a lot of people who want to do experiments on non-human primates leave the US and are conducting them elsewhere that have different regulations or requirements for that kind of research, where in the US we have very strict and necessary regulations requirements to work on animal models.
Okay, thank you. We do have a question about how should policymakers view collaborations and cooperation with biotech research with worldwide, especially those we have more cooperation with and those that are strategic competitors.
I can make some general comments, but I think it's a good thing. I think we live in a connected world, and I think that expertise is not always going to be in the United States. And you could just look at the COVID vaccines and the expertise that was at Biotech that partnered with the U.S. company in order to give us our first COVID-19 vaccine. So I think that it should be viewed positively. I think that there were some, in some of the policy discussions, they wanted certain things to be made in, have a made in a USA clause type of thing. I don't think that makes sense because we don't know where the talent is going to come from. And I think we have strategic partners that have great talent. And I think that when you're making these types of decisions, I think they don't necessarily have to be all related to domestic companies. I think that it's important to not restrict the funding that way, because I think, again, as I said, it's not something that's always only going to have the expertise in the United States.
Okay, thank you very much. Okay, I think we have one more question about non-human primates and whether recent developments in policy towards them is impacting the research and our work in a detrimental manner.
There's no question that use of non-human primates have declined in the US. And we know that also importing non-human primates, particularly from China, where 60% of the world's non-human primate use in research came from China, and they have restricted all exports of monkeys. So I mean, that's a good policy question that requires a lot of discussion about, animal models. It's definitely declined in the European Union as well. And we need to have that discussion about the trade-offs between using and not using animal models, especially non-human primates.
Okay, thank you. We have a question about the breakdown in manufacturing again of where these are and what's the breakdown between the US and foreign. You can speak to that.
Sure. So in the bigger scheme of things, we have about a third of these medical countermeasures have no domestic manufacturing, meaning that we import that directly. And it's an important thing to think about redundancy. So, if all drugs are made in only a couple of facilities and something happens to that facility or there's a contamination, et cetera, those drugs are no longer going to, be able to be distributed or made. So we're providing this data for policymakers to have that information and making decisions for how to reshore manufacturing for these drugs. But again, it's important to think about where we get the materials to make these drugs, and then how are we making with our workforce.
Okay, thank you. I think we have time for one more question for both panelists. And the question is, what would be the first step if we wanted to move down this path of broadening research for the viral families?
To me, I think the first step is for the way funding in pandemic preparedness is done from a congressional standpoint to explicitly be open to viral family approaches when they're funding BARDA, when they're funding NIH, that it's not so much tied to this funding has to be on, as I said, this specific virus, allowing those funding agencies to broadly fund projects in those viral families, even if whatever the researcher is doing is not necessarily in and of itself, a highly regarded pandemic pathogen. That, I think, would be the way to do it, is to just broaden that and not have it so earmarked towards specific viruses, so that it can be, so those agencies can work proactively in those viral families and do a lot of research that can be repurposed for pandemic preparedness issues.
I 100% agree. It has to start with that basic research and how we incentivize, fund, staff, researchers in these specific viral families. But we also need to make sure that we take those great discoveries and bring that into the clinic and bring that to patients. So it's really taking it from the very beginning of the scientists creating this knowledge and these drugs, to being able to make that into safe clinical trials, and then also scale up that production to bring it to patients.
Okay, great. Thank you so very much. I'm going to turn it back over to Lynn.
Thank you. And on behalf of CSET, in addition to our moderator, Dr. Anna Puglisi, I'd like to thank co-panelist Dr. Caroline Schorger, and most especially our guest speaker, Dr. Amesh Adalja. Thanks as well to everyone who joined us today. We appreciate your interest and the many thought-provoking questions, and we're sorry that we could not get to them all. If you'd like to learn more about CSET, please go to cset.georgetown.edu and sign up for our newsletter and research updates. Our next event takes place October 19th and will feature CSET's Tim Huang and Emily Weinstein, together with Martin Rasser of the Center for a New American Security. It's called Decoupling in Strategic Tech Sectors, and it addresses what the satellite industry can teach us about future efforts to separate technology supply chains. In the meantime, look for our email tomorrow with follow-up info from today's event, including details on where to find the video recording. Take care, and we hope to see you again real soon.